This is Bio 181, Chapter 3, the key concept for the macromolecule proteins. So proteins have many different functions. They're the most diverse in terms of their function. A key category that's primarily made of protein are enzymes. 99% of the time, enzymes are proteins. We had a whole chapter basically about them. They are biological catalysts. A catalyst is something that speeds up the rate of a reaction. And the monomers are called amino acids, and the polymers are called polypeptides. Okay, so the monomer is an amino acid. Amino, that word root refers to the fact that it has nitrogen in it. An acid is the fact that it donates the hydrogen to solution. So remember, acids are going to increase the hydrogen ion concentration of a solution. So they readily donate hydrogen to solution. All right, so that's where it gets its name. The amino acid, the basic structure of the amino acid is we have a central carbon, and then remember that carbon forms 4 bonds. So on its outer valence of the orbital, it has 4 electrons that are unpaired. So it can pair up with 4 different atoms. One of the bonds is just with hydrogen, so that's simple. And then it's also going to be with an amino group, which is NH2. This is a functional group. That's one of the bonds. Another bond is to a carboxyl group, which is C double bond O, and then an O to an H. Okay. So this here on the right is carboxyl. This is a functional group that we learned in a previous chapter. Over here on the left is another functional group. This is amino. And the carboxyl group is going to be the one that gives it the name an acid because it readily donates this hydrogen. That will often pop off, and this will have a negative charge over here. Now, interestingly, over here, this amino, if you remember the characteristic, the chemical property of an amino group is that it will actually take up pretty readily a hydrogen out of solution. And so this, if it takes it up, will actually have a positive charge. So this molecule can donate hydrogen, so it acts as a weak acid, and it also acts as a weak base, depending upon the solution. And so that means that itself, the amino acid will act as a buffer. So all proteins act as a buffer in an organism, and it's one of the primary buffering systems that keep an organism from becoming too acidic or too basic. So we're still missing, I'm going to go back to this carbon here in the center. So we looked at these bonds. We have one more bond to look at, and that is bonded to what we call an R group. And this R group can be multiple different things. There's about 20 different R groups, so 20 different types. And some of them are going to be neutral, or they can be charged, or they can be polar. So there's the variety here. This R, I always think of this as this is the remainder of the molecule. And there's 20 different types of amino acids. Each of them has a different R group. Okay. This is just an image of the amino acid. It's a little bit neater than my drawing. And you can see over here the amino group on the left, the carboxyl group on the right, our hydrogen on the top, and then this R is the side chain. which could be something as simple as CH3, which would be a methyl group, which would mean this is a relatively non-polar example, or it could be something that's charged or polar. Okay, a polymer is multiple amino acids put together. So again, the monomer is the amino acid. So if we have a whole bunch of these put together, right, thousands of these potentially, then we have what would be called a polypeptide. So poly is many, so polypeptide. Why that O is red, but it is, so polypeptide. And all of these bonds here, these are covalent bonds. And the bond has the name of a peptide bond. And so that's where we get the name polypeptide for this polymer. So again, it's just a whole bunch of amino acids put together. And then depending upon the R group on these amino acids, some parts of the molecule are going to say be positively charged, and some parts are, if the R group say is negative or negatively charged. And so they'll actually interact and they'll often fold back on each other. So there's actually 4 levels of protein structure. These are very complex molecules, or at least they have the capacity to be very complex molecules. 4 potential levels of structure. So the first one is primary, and that's just the amino acid sequence. So the amino acid sequence. And so basically we just have a sequence of amino acids. Okay, that's the primary structure. The secondary structure, the second level, is where if this is a very long molecule, it can fold back and interact with itself. And so say there's a whole bunch of molecules over here, and they can be hydrogen bonded to another part of the molecule. That would be the secondary structure, and there's 2 main types of secondary structures. There's what we call alpha helix, which results in sort of a spiral structure and looks like a spring when it's diagrammed. And then there's a beta pleated sheet, and that sort of looks like a corrugated. A corrugated roof or something, that kind of has this accordion-like shape. And then, oops, between those, there are hydrogen bonds. So those are the 2 types of secondary structure. The third level of structure is called tertiary. It's the third level. And the tertiary structure is going to be if we have covalent bonds that form between different parts of the molecule. So say you've got an alpha helix here, and then you've got a beta pleated sheet, and then you have another alpha helix, and then another beta pleated sheet. If between these R groups that are on every monomer, there's all these R groups. And so some of these R groups can interact by forming a covalent bond. If that happens, kind of small down here, so I apologize for that, then we're going to have a really strong bond. So a covalent bond is much stronger than a hydrogen bond. So, and this is generally going to form, if you remember your functional groups, there's a functional group called a sulfhydryl. And that sulfhydryl, the characteristic of that is that it will interact with other sulfhydryl functional groups and create this covalent bond. So between the 2 sulfurs, there's going to be what's called a disulfide bridge, and that will actually be very strong and link this tertiary level. The last part is only found in some proteins, and that's called quaternary. So the fourth level, remember this is 4 levels, that's quaternary. And that's if you have multiple, oh gosh, my phone's ringing. Okay, let me turn that off. Okay, so multiple polypeptides. And with multiple polypeptides, so say you've got one structure over here and it's got alpha helixes and beta pleated sheets and alpha helixes and beta pleated sheets. And then they're bonded with covalent bonds to each other. So you've got, here's one polypeptide, and then you've got another polypeptide over here that has some alpha helixes, and maybe a alpha helix here, and then a beta pleated sheet, and then an alpha helix, okay? And then there's a covalent bond between this. If these two interact, so now this polypeptide is going to be bonded to this polypeptide. So maybe there are some hydrogen bonds between it and maybe covalent bonds between it. Now that is the quaternary structure. All right, and that's it for proteins.